James Arima. Well, it looks like we are having a challenge. So, anyone else? James and Brian, your mics are on, so I thought maybe you wanted to contribute to the issue. I'm asking anything you can remember from the last lesson that we had, or the last 3 lessons that we had last week. Okay, Josephine.
Hi everyone.
Hi.
I can remember the definition to biology and the branches, the importance of studying biology,
Yeah.
and we also learned about the double naming. We learned about the microscope, that is the light microscope. Yeah. So, do I go bits by bits or?
Just choose one area and just talk briefly about it, and then yes, just one area of the ones that you've mentioned.
Okay, I'm gonna talk about the double naming. And
Go ahead.
double naming, we said that the double naming process is called binomial nomenclature, where the scientific name the scientific name is written in capital letters, and the species name is written in small letters. And then when it is typed, you write them separately, and you underline them separately. Okay.
Okay.
Yeah.
That is good. That is very good. So just like she has said, let me do repeat ups. We said that binomial nomenclature is the scientific double naming. That is very correct. We also went ahead and said that when you write a scientific name, the first name is usually the genus, and it starts with a capital letter, and the species name is written in small letters. So that's very good. Thank you. Thank you very much. So anyone else who has something to say on what they can remember, kindly? Yes, James. Well, looks like we'll have to continue then because of time. But thank you, Josephine. So just like Josephine has said, we talked about a lot of things, beginning from introduction. We went ahead and talked about branches of biology. Went ahead and talked about importances of studying biology in high school, characteristics of living organisms. We also talked about collection of specimens. We looked at the tools which are used to collect specimens and stated their functions. We also went ahead and talked about differences between plants and animals. We talked about classification 1, where we grouped organisms based on their characteristics, that is the similarities and differences. Talked about the taxonomic units. We went ahead and talked about magnification, and we learned how we can calculate magnification. We gave rules that we follow when we're doing biological diagrams. Talked about taxonomic units of classification, that is kingdom, phylum, class, order, family, genus, and finally species. And here we say that the kingdom has the largest number of organisms and the species is the smallest one. So, and we also say that organisms in the same species can easily reproduce and give rise to an organism that is capable of reproducing. So we talked about kingdoms of classification. We talked about scientific naming, the one that we have discussed with Josephine. We also went ahead and talked about the microscope. and we said that we have two types of a microscope. That is the light microscope and electron microscope. So I told you to look at a microscope in your free time, so that during today's lesson we can talk about a microscope, parts of a microscope, and the functions of a microscope. And so the lesson begins. So a microscope is an instrument that is used, or an apparatus, but usually we say an instrument. It is used in hospitals, it can be used in labs, it can be used in schools, but basically the function of a microscope is to magnify or to make organisms appear larger than they are. Usually, some microorganisms are so small. The reason why we call them microorganisms is because they are so small that we cannot see them with our eyes. So therefore, what do? We have a microscope which we use to magnify those small organisms and make them appear larger, so that we can be able to see them, study their parts, and see how they even function. So parts of a microscope, like you can see here, there are very many. There are around 12 parts of a microscope, and each and every part that you can see here from the display has a function. So just before you even talk about the functions of a microscope, I want us to talk about the parts. So on top here we have on top here we have that part there which we're calling the eyepiece. Now from the word eye, from the name eye, this word eye here, so we get the name eyepiece. Okay, so eyepiece meaning we use an eye in this part. So when we're looking at, sorry, excuse me, there's someone's mic that is on. Marriott, kindly, your microphone, thank you. So as I was saying, this part here, the eyepiece, comes from the word eye, which means we are using our eye on this part. Okay, so when you're observing anything in the microscope, you observe through the eyepiece. So this is where you place one eye. Of course, it's only one eyepiece. We use one eye. So you close one eye and you use the other eye to observe. Now, this eyepiece part here usually has a lens that magnifies the specimen. Okay, so it has a lens that magnifies the specimen or contributes to a specimen. Now, those people that are maybe wondering what's a specimen, like we said in the last lesson, a specimen is either a part of an animal or a plant, whole of the organism that we place on a microscope or we study in a lab. So when you take an organism or part of an organism and you study it, it changes from being an organism. Like for example, if you're studying a grasshopper, it changes from being a grasshopper, and since we're using it in a biological class, it becomes a specimen. So when I mention the term specimen, I mean either part of an organism or whole organism. So when we place an organism on the microscope, we will observe it through the eyepiece. This is because the eyepiece has a lens that is used for magnification. Okay, then we have a body tube. That part there, we have a body tube. Now a body tube holds this part here we are calling the objective lens. That part there, the objective lens, it is held by the body tube. And a body tube is hollow. It doesn't have a lens. It is just hollow. So it holds the objective lenses and the eyepiece. So if you look at the body tube, it is the part between the eyepiece and the objective lenses. So body tube's function is to hold the eyepiece lens and also to hold the objective lenses. Now, let us look at the objective lenses, this part here, the objective lenses. Now, objective lens, it means this part here has a lens. Now, which other part has a lens apart from the objective lens? It is the eyepiece lens. Are we together? So therefore, eyepiece lens and the objective lens are both used for magnification. So an objective lens is a lens that magnifies, and then the eyepiece is also a lens that magnifies the specimen. So how do you know how many times a specimen has been enlarged or magnified? You check, usually at the eyepiece part here, you can see the number of times that eyepiece is capable to magnify. So usually inakuanga imeandikwa maybe times 10 ama times 5 ama times 20 ama imeandikwa times 50. So it depends on the type of microscope which you are using. So based on the type of microscope you're using, the power of the eyepiece lens then it becomes. So for example, if you want to observe very small microorganisms, you need a microscope that has a very high magnification power in the eyepiece lens. So kama tuseme you want to observe something as little as a bacteria, unaweza pata the eyepiece, it has a magnification of maybe times 500. So what that means is the the specimen has been made large 500 times. So no matter how small it is, 500 times is a high number of enlarging that specimen, and you can comfortably see that specimen. So we have said we have two types of lenses in a microscope. The first lens is found on the eyepiece, and the other one is found on the objective lenses. So when these two eyepieces combine, they make the specimen to appear bigger. Okay, so eyepiece lens and objective lens, they are the ones that are used for magnification. I hope you have understood that. Good. Then we have the body tube. The body tube holds the eyepiece and it also holds the objective lens. Okay, now there's a part here which is called a revolving nose tube. Revolving comes from the word kuzunguka. Revolving means ni kuzunguka. So revolving nose piece is used to switch objective lenses. Now, objective lenses haikuangi moja. It is just three. There are three lenses. So hizi objective lenses, they are three. So these three lenses are different even in size, in shape, and the number of times it can magnify a specimen. Now we have three types of objective lenses. We have one which is bigger. So if you look at them, maybe it is bigger. Then we have another one which is a bit smaller, and then we have a smaller one kabisa. So those are the three lenses. Usually there are three. We have the larger one, the medium one, and we have the smaller one. So these three lenses are named based on how many times they can magnify a specimen. Like for example, the smallest lens of them all, that one, it is referred to as a low power objective lens. Low power objective lens. What that means is this lens can magnify specimens using, actually it is the lowest, okay? Of the three, it has the lowest magnification. Like for example, if this one had times 10, the second one would have times 5, and then maybe the third one would have times 2. So here katikati ikuwe times 5, alafu the high power. So we have low power ikuwe na times 2, it is the smallest, followed by the medium power. Medium power means it has a higher magnification than the low power, and it has a lower magnification compared to the big objective lens. So objective lenses are three. We have a low power. Then we have the second one, the medium one. This one, we call it a medium power. Medium power. And then we have a high power objective lens. So types of objective lenses are number one, low power. Number two, we have objective lenses with a medium power, and then finally the third one, it has a high power magnification. So usually high power iko na magnification mingi, followed by the medium power, and then the low power. So how do they work? This is how they work. If for example you have a very small specimen. Kindly, Kate, Maura, your microphones are on. Brian, your microphone is on. Kate, kindly switch off your microphone, sorry. Thank you. So as I was saying, let me repeat that kindly. I was saying, how do objective lenses function? Objective lenses function together with the eyepiece. So if, for example, you have a very small organism like a bacteria, which is so small, you can't even be able to see it. And even if you made it larger like 2 times or 3 times, you cannot be able to see it. So at the eyepiece lens, you place the highest lens you have. Usually you can find a lens that is maybe times 10, it can be times 15. That is at the lens, okay? Then at the objective lens, we can have maybe the low power being times 2, then the medium power being times 5, and then we have the high power being times 10. So if you want to magnify a specimen and make it as large as possible, you take the highest magnification power yenye ni high power, which is times 10, and then you use the eyepiece lens, which is going to be times 15. So what will be the total magnification? It will be times 15 of the eyepiece lens multiplied by times 10, which means the magnification will be times 100. Okay, so depending on how small a specimen is, that is when you choose which objective lens to use. So if a specimen is big, like a grasshopper, and you only need to magnify it just a little bit, then you're going to use the low power objective lens. So the purpose of having 3 objective lenses is so that you can switch between, among the 3 lenses based on how large you want your specimen to be. Now, that is where the revolving nose piece comes in. So as you switch from one objective lens to another, then you will be using a revolving nose piece. So a revolving nose piece inasaidia objective lenses zizunguke, una change from one objective lens to another. So I'm going to repeat, I've said the eyepiece lens is going to be used in magnification because it has a lens. Then the body tube is going to hold the eyepiece lens, and it actually holds the objective lens, both of them. So without the body tube, we cannot have the eyepiece lens and the objective lens. And then I've said objective lenses ni tatu. We have the low power, we have the medium power, and we have the high power. Is very small, you're going to use high power so that it can be magnified more times. If you require to observe a specimen that is neither small nor is it that big, then you're going to use a medium power because you only need to magnify it just a little bit. And then if you have a specimen which you can actually be able to see and handle and even touch, you then use a low power, the smallest one, because you only need it to be magnified just a little bit. So those are the functions of the eyepiece lens, the body tube, and the objective lenses. And switching from one objective lens to another is facilitated by the revolving nose piece. Very good. From there, let us move to the coarse adjustment knob and the fine adjustment knob. So these two parts here are very important in a microscope. You see, this part which you're calling the body tube, the one that holds the eyepiece lens and the objective lenses, it moves, okay? It can move either up or it can move downwards. When it moves upwards, it makes the objective lenses to move away from the specimen. Now let me show you where a specimen is placed. If, for example, you want to observe a specimen in the lab, you place it here on the stage. Okay, so you take your specimen, you place it on the stage. Na hapo kwa stage you can see there are clips. Okay, the function of clips, there are usually two, but here they have labeled one, but there's another one on the other side. So clips hold the specimen in position. So you see, like the way you would place something on the table and then unaona inaanguka unashikilia. So we use a clip, like the way the lady's hair is usually held by a clip when they plait your hair or they make their hair or blow dry their hair. So those clips usually look almost like those ones, but they are a bit smaller. And their function is when anyone who is using a microscope places a specimen on the stage, hapo juu hivo, then the clip is used to hold the specimen in position. So kazi ya clip ni kushikilia a specimen on the stage. Okay, so when you place a specimen on the stage does not move. The only part of a microscope that is able to move, it is the body tube. So it can move upwards or it can move downwards. So when it moves upwards, it moves away and further from the specimen. When it moves downwards, it will be moving towards the specimen. So another way of making sure that you're able to observe your specimen very well, what you do is you will move the body tube closer to the specimen or further away from the specimen so that you can be able to adjust the magnification. So for example, if you're using a high power objective lens, it means that this high power has a very big lens, a lens which is able to magnify the specimen even very many times. So that means you can move the body tube up a little bit so that the lenses will be able to observe what is on the stage. If for example you're using a low power objective lens, meaning that lens has lesser power, then you can move the body tube closer to the stage. Okay, how do you move the body tube closer or further away from the stage? You move it using the coarse adjustment knob. So you use a coarse adjustment knob to move the body tube along the longer distances. Okay, so if you want it to move further away from the stage or closer to the stage, you use a coarse adjustment knob. So coarse adjustment knob moves the body tube across very long distances. Okay, so inaweza anza kutoka hapo ikuje huku juu kabisa. So it depends on the distance which you want to move it. So bottom line here is the function of a coarse adjustment knob is to move the body tube across longer distances. Okay, after we've talked about the coarse adjustment knob, let us talk about the fine adjustment knob. From the word fine, it means that the adjustment knob moves the body tube, yes, just like the coarse adjustment knob, but it moves it across very small distance. Okay, a very short distance, I mean. So it only moves it in millimeters or inches. What is the essence of moving the body tube across very short distance? This one brings clarity on the specimen which is on the stage. It's just like the way you use a camera. For those people that have used a camera before, sometimes a camera zooms and brings an image closer to you, and then sometimes you have to move the camera so that it can make the image to be more clear. So like the way you tap on the screen when you're taking a photo so that the photo becomes clearer on the phone. So when you use a fine adjustment knob, it moves the microscope across very short distances, and the purpose of using a fine adjustment knob is to bring clarity or bring the image into a very sharp focus. So here image inakuwa very clear that you can be able to see what you're observing and you can actually be able to draw because it has been made clear by the fine adjustments knob. So before I continue, let's go back to the first one. So we said the first part of a microscope which we have learned today is the eyepiece lens. And we have said the eyepiece lens has a lens that causes magnification. Okay? That lens which is found in here is used together with the objective lens to magnify the specimen. So, unaangalia objective lens iko na power inatoshana aje. Maybe you find it has a power of times 10 and then the eyepiece has a power of times 5. So here specimen iko kwa stage, ukiangalia itakuwa imemagnifyiwa ama imefanywa kubwa mara 5 plus mara 10 za objective lens. So 10 times 5 will be 50, which means that specimen will have been enlarged 50 times. So I've said the eyepiece lens has a lens that is used for magnification, and then we have an objective lens which has a lens which has lenses which are used together with the eyepiece for magnification. We've also gone ahead to say that we have 3 objective lenses. That is the low power objective lens, that the other one is the high power objective lens, and we also have the medium power objective lens. Nikasema high power has the highest magnification. That's that kama tuko na lenses tatu, tukasema ya kwanza yenye ni high power iko na power kubwa, so it enlarges the specimen even more compared to the rest. Then we have the medium power, which is not that, it doesn't have that much power for magnification, but it is not the one that has the lowest magnification. And then we have the low power objective lens, and it has the lowest magnification. And even when you look at these objective lenses, they differ in size. High power is the largest, followed by the medium power, and then the smallest is usually the low power. We've also gone ahead and we've said, for us to be able to switch from one objective lens to another, because there are 3, we use the revolving nose piece. So the function of a revolving nose piece, we say it allows changing from one objective lens to another. That is very important for you to note. And then we've said there's a part here which is known as the body tube. This body tube part here, we have said it holds the eyepiece and also holds the objective lenses. So from there, we've talked about other 2 very important parts of a microscope, and that is the coarse adjustment knob and the fine adjustment knob. But we have said the coarse adjustment knob is the largest, and its function is to move the body tube across very long distances to bring the objective lenses closer to the specimen or further away from the specimen. Because we have said a specimen is usually placed on the stage for observation and it is held by the clips. So when you place the specimen there, if you bring the objective lens closer or the body tube closer to the specimen will appear bigger. Ni kama venye tuseme umekaribia kitu, unaiona ikiwa kubwa. Ukipeleka mbali, unaona ikiwa ndogo. So, the function of the coarse adjustment knob is to move the body tube across very long distances so that we can have magnification taking place easily. And then we have talked about the fine adjustment knob. Fine adjustment knob is smaller than the coarse adjustment knob. And the function of the fine adjustment knob is to move the body tube across very small distances or short distances, so that we can have a very clear image. So what is the function of the fine adjustment knob? Moving the body tube across short distances and to bring the image into a sharp focus. To bring the image into a sharp focus. That is the function of the fine adjustment knob. Then we have another part here, which we're calling the arm. So there's that part there, which you're calling the arm. And this part here, which you're calling the arm, has a very simple function. This is where a person or whomever is operating on a microscope holds while either moving a microscope or while holding the microscope. So if you decide to move a microscope from one place to another, you hold it by the arm. So an arm can also be referred to as a limb. So this is a part of a microscope which is used while moving the microscope. So the person operating the microscope holds the microscope on the arm, whether they are working on the microscope or whether they are moving the microscope from one place to another. So then from there we have the stage here, but we had talked about the stage and we said the stage is where a specimen is placed. So if you want to observe anything on a microscope, you place it on the stage. When you place it on the stage, you are then able to move your lenses on the body tube and you'll be able to see the specimen even more better. Then from there we have the clips. We have clips here and we've said clips function almost the same way like the clips that we use for ladies' hair or clips that we use to clip on files. Their function is to hold. So clips hold on to the specimen. So when you place a specimen on the stage, you use the clips to hold on to the specimen so that it doesn't move up and about the stage, so that when you even move the microscope, the stage, the specimen on the stage remains intact. Okay. From there, I want us to look at this part here, which we're calling the mirror. Now, one thing we should note is this equipment we are seeing here is known as a light microscope. A light microscope uses light, definitely. It uses light. So if it uses light, where will the light come from? The light comes from the sun, okay? So there's light from the sun. And when that light hits the mirror reflects that light, okay? I'm saying light from the sun is reflected by the mirror and it is reflected inside the microscope so that it can light up the specimen and we are able to see the specimen. So what is the function of the mirror? To reflect sunlight, okay, onto the specimen so that we can light up the specimen on the stage. So the mirror's function is to reflect sunlight onto the microscope, into the microscope, so that we can be able to light up or illuminate a specimen. Sawa sawa. That is the function of the mirror. From there, we can talk about another part of a microscope, which is diaphragm. Okay, diaphragm. Now, diaphragm can easily be opened and closed because the function of a diaphragm is to regulate how much light passes through. You see, sometimes we can be observing a specimen in areas where we have excessive sunlight. And when excessive sunlight passes through the microscope, it might affect the way we see the specimen. So to control how much light passes through the specimen, from the sunlight which is going to be reflected by the mirror, then we can easily open or close the diaphragm. So a diaphragm can close so that it is very small and it can even be opened as wide as this. So when you want a lot of light to pass through, you open the diaphragm, allowing sunlight to pass through. When you do not want light to pass through, then you close the diaphragm so that light does not pass through in large amounts. I want to demonstrate what I mean using my hand. So let me switch the camera on so that I can demonstrate what I mean. Okay, so give me a minute. So let me see if I can be able to do this. Good. So a diaphragm, I hope you can see. A diaphragm looks something like this hand that I'm showing you here. So a diaphragm has the ability to be either opened up widely or to be closed up. So when you're using a microscope, there's a way you can actually allow whichever amount of light you want to pass through. So let's assume we are observing a specimen using a microscope and we are in a room that does not have enough light. So that means we need a lot of light to pass through. So what you do is you open your diaphragm as wide as possible to allow as much light to pass through as possible. When you are in a room that has enough light, and sometimes the light can even be excess, you close the diaphragm to leave a very small space for little or less light to pass through. So the function of a diaphragm is to regulate or control the amount of light passing onto the inside the microscope. So diaphragm is used to control or regulate the amount of light passing to the specimen so that it can light up the specimen. So I hope you have understood the concept of the diaphragm and how it functions. So if you need too much light, you open it widely. If you do not need too much light, what you do is you close it, and when you close it, it becomes very little light is going to pass through. So that is how a diaphragm of a microscope functions. So I hope you have understood. Good. So we continue on to the other part of a microscope. We have a stand. We have a stand. So let me go back to sharing the notes or a base. Okay, stand or a base. And a base is basically where a microscope uses to actually get support. So when you're placing the microscope on a bench, you place it on the side of the base, or what we call the stand. So let me get to the note. Good. So we have the stand or we have the base. This is the stand or the base. This is where we place our microscope on the bench on. So tukiekelea kwa bench, tunaiwekelea kwa base. So what is the function of the base? Base of a microscope is used to provide support to the microscope. Okay. So we place it on a stand or a bench so that we can so that it can provide support. So actually in an exam, when they ask you what is the function of a base or a stand, you'll say it provides support to the microscope. It provides support to the microscope. So far we've talked about the fine adjustment knob, we've talked about the body tube, the coarse adjustment knob, the fine adjustment knob, revolving nose piece, objective lens, stage, clip, hinge screw bado, but we've talked about the arm and the base and the mirror and the diaphragm. Now the parts which I want us to talk about now are the hinge screw. Now hinge screw is easy because it is used to actually hold the microscope parts together. Okay, so these microscope parts are used to they are held together, especially around the base, using the hinge screw. Then we have a condenser, this part here which you're calling the condenser. When light passes from the sun, it goes to the mirror and it gets reflected. So light from the sun is going to be reflected by the mirror towards the microscope. When it gets to the diaphragm either closes or opens to allow the amount of light which you want. But for the specimen which is on the stage to have enough light, that light is directed by the condenser. Okay, the function of a condenser is to concentrate light to the specimen on stage. So that light is directed to the stage by the condenser. Okay, so if this is the sun, like that, the sun rays will hit the mirror like that. Then the mirror will reflect the light, and based on how much light it is, you will be the one to regulate the flow. So if you want too much light, you open the diaphragm. If you want less light, you close the diaphragm so that very less light will pass through. Once light has passed through, it moves to the condenser where it will be directed to the specimen on the stage. So the function of a condenser is to concentrate light to the stage for illumination. Illumination means lighting up, so that we can have light lighting up the specimen, and then you can use the lenses for observation. So usually in an exam, they can either draw for you a microscope and tell you to name the different parts of a microscope, okay? Or they can give you a part of a microscope and they ask you to give the function of a microscope part. Are we together? So therefore, let us now look at the notes on the parts of a microscope. So the first part is the limb or the arm, where we said it supports the body tube and stage, and this is also where you hold when you're moving the microscope. Then we have the base or the stand, where we said it provides a firm and stable support to the microscope. This is where you place your microscope on the bench using so that it can be given support. Then we have the body tube, and we have said the body tube holds the eyepiece, the revolving nose piece, and the objective lenses. Then we have the eyepiece, which we have said it contains a lens which contributes to magnification of the specimen. So that lens is used for magnification. Then we have a coarse adjustment knob, where we've said it raises and lowers the body tube through long distances and brings an image into rough focus. It brings the image closer to the objective lenses. Then we've talked about the fine adjustment knob, where we said it moves the microscope or the body tube along smaller distances and it brings the image into a very sharp focus so that you can be able to see. Finally, we have the diaphragm, which you've said it regulates the amount of light passing through the condenser to illuminate the specimen or to light up the specimen. And then we've talked about the objective lenses, where we've said an objective lens has a lens that is used in magnification, because we've said it's only two parts of a microscope that have a lens, that is the objective lens and we have the eyepiece lens. So both are used for magnification or to enlarge the specimen. Then we have a mirror. A mirror reflects light through the condenser. Tumesema it is going to reflect light from the sun so that that light can be used to illuminate or to light up the specimen. Then we have the revolving nose piece, where we've said it enables changing from one objective lens to another, because we have said we have three objective lenses, that is the low power, the medium, and the high power. Very good. So then we have a condenser. We have a condenser which you've said its function is to concentrate light onto the stage. So light is not diverted or diverged into different places. No, it gets converged onto the stage peke yake. So the function of a condenser is to concentrate light on the stage where we have the specimen. And then we said the stage is a platform where a specimen is placed. And usually when using a microscope, we do not just place a microscope alone. A specimen on the microscope. Usually we use a glass slide. So a glass slides is usually like that. So that slide is usually a piece of glass that has been cut in a rectangle shape, and then you place your specimen on that. Let me use a different color. You place your specimen on that slide like that, and when you place it there, you use the microscope to observe and enlarge that specimen. So a slide is a rectangular glass piece which has been cut, and this is where we place a specimen. So we don't just place a specimen on top of a microscope. It's usually on a glass slide. So it is the glass slide that we take and place it on the stage of a microscope for magnification. And then we have the clips. When you place a slide on a stage, the clips actually lie on top of the slide like that, and they hold the slide in position. So it holds the slide in position, the slide that contains a specimen. So this is the specimen. and this is the slide. Okay, yes, and then of course these are the clips. So the clips are going to hold the slide into position. So those are the different parts of a microscope. I hope and I want to believe that you have understood about the different parts of a microscope. So is there any question on the parts of a microscope and their functions? If there is, kindly ask, because we are about to move from that particular subtopic and move to something else. So is there any question? Is there any question on parts of a microscope? If there's a question, just ask. If there isn't, type understood so that we can proceed. So kindly give response so that we can proceed. If you have understood, type I've understood. If you have a question, it's your chance to ask the question. So good, understood, understood. That is good. Wonderful. Now that we have understood, I want to believe that we are not going to have a challenge answering a question. After the lesson, I'm going to post a few questions on this particular subtopic in the group so that you can go through them and see how you should have answered the questions in case they appear in an exam. And also, if you get a challenge with a question, you can always ask those questions or you can reach out to me and I'll be glad to assist you. Good. So from there, I want us to move into something which we call an electron microscope. Okay. There are two types of a microscope. Usually, we have a light microscope which uses light to illuminate the specimen. Tumesema jina illumination means to light up ama kumulika. Okay. So light microscope uses light to illuminate the specimen. So in an exam, if they draw for you this particular apparatus or equipment, alafu akuulize name the above drawn apparatus, you should say it is a light microscope, because we have two types of microscopes. We have a light microscope and we have an electron microscope. A light microscope uses natural light, probably from the sun, okay? But an electron microscope cannot use natural light. An electron microscope uses beams of electricity, ama electron beams. What that means is you must plug an electron microscope into a socket that has power so that that socket will provide light to illuminate the specimen. So you can use a light microscope while you're using sunlight, but you cannot use an electron microscope using sunlight. It must be connected to power so that you can be able to observe anything under an electron microscope. So let's talk about then differences between an electron microscope and a light microscope. Differences between an electron microscope and a light microscope. Good. Difference number one. Okay. Light microscope uses light to illuminate the specimen. So this one uses light from the sun, light from bulbs, light from any source of light, any natural source of light. But for the electron microscope, it only uses beams of electricity or electrons to illuminate the specimen, meaning it cannot be operated without power. Then we have light microscope uses glass lenses for magnification, while electron microscope uses magnetic lenses for magnification. Why are we saying that it uses magnetic lenses? It is because magnetic lenses work with electricity or power for magnification. But since a light microscope uses natural light, we just use glass lenses for magnification. Okay, lenses ni kama zenye watu, for example, ukivaa macho nne, those that wear those glasses. So usually in the glasses we have a lens that maybe if you have a challenge uh, with seeing small digits, they magnify. So usually in a microscope, light microscope to be specific, those lenses are like the ones that people wear wakivaa macho nne, yenye inakuanga na ability to magnify something or make it appear bigger. Another difference between a light microscope and an electron microscope is that a light microscope has low magnification. It has low magnification power, meaning if you want to see a very small organism, your best bet to see it is not a light microscope. A light microscope will not make a specimen appear very big. It is used to observe specimens which are roughly, at least they are not as small as you will. That is why when you sit and you go to the hospital, they ask you for blood or stool or saliva, and then they run it under an electron microscope because it can be able to see even the smallest of the microorganisms. So the difference here is a light microscope has low magnification power, while an electron microscope has a very high magnification power. Okay. Another difference is that a light microscope can be used to view living or dead specimens. Okay, so when you're using a light microscope, you can use a dead specimen or a living specimen. But for electron microscopes, you can only use dead specimens. Okay, you cannot observe uh, a live specimen using an electron microscope. The other difference is that in light microscope, the specimen is stained using normal dyes. Now there's a concept which has come up here, staining using dyes. So someone is asking, what is staining? Now staining means you take a slide that has a microscope and then you pour a dye on top of that specimen. A dye includes inks such as inks and some chemicals such as iodine. I don't know if you know iodine. Iodine is I'm sure at some point you have seen someone who has had an injury. Maybe they've cut their hand or maybe whichever part of their body. When they go to the hospital, the doctor uses some orange, almost orange liquid to clean their wound. That is what we call iodine. So in microscopy or when you're observing a specimen in the lab, we use an example of iodine. Iodine now becomes a dye. You see, when you look at it, it has a brown, almost a brown color, right? So that brown color makes the parts of a specimen to be visible. Okay, so the process of applying that dye or that iodine on a specimen which is on a slide, we call it staining. That's what we are saying. In electron microscope, we do not use normal dyes. We stain using complex dyes. But for light microscope, we can use normal dyes such as iodine. So in an exam, one way in which they type these, they set this question from this area is that they just ask you list three differences between a light and an electron microscope. When they do that, the first difference you should give is on magnification power. An electron microscope has a high magnification power compared to a light microscope. The other difference is a light microscope uses light to illuminate specimen, while an electron microscope uses beams of electrons to illuminate the specimen. The other difference is under a light microscope, the specimen under view can be living or dead. So you observe both living and dead microscope. While in an electron microscope, the specimen under view is usually dead. So you cannot observe a living specimen under an electron microscope. So I believe those are the easiest 3 or 4 differences. But on top of that, you can add an electron microscope has a low resolving power, a light microscope has a low resolving power, while an electron microscope has a high resolving power. Now, question is, what is resolving power? Resolving power is an ability to distinguish two parts of a specimen. Now, I'm sure you have come across terms like conflict resolution or resolving conflicts. This is where two people are disagreeing. Maybe they have a conflict, and then you listen to their case, and then you can help them to resolve the conflict. This means these two people have differences, so you just provide a clear cut solution to those particular differences. So therefore, even in a microscope, sometimes you can have a specimen that is so small, you cannot be able to distinguish between a nose and also the mouth, or maybe even the other parts. So most of the parts are very close to one another. You cannot be able to distinguish them. So what you do at that point is you look for a microscope that is so powerful it can enlarge that specimen to an extent that you can be able to see the different parts despite the fact they can be close to one another. So when we say that an electron microscope has a higher resolving power, we mean you can be able to tell of different parts of a specimen or different features of a specimen easily, because it is going to magnify the specimen even much to magnify the specimen more than a light microscope can. And since a light microscope can only magnify a specimen to very not a lot of power, maybe 50 times, 100 times, so it's very hard to have a resolution to see the different parts of a specimen because the magnification power is low. So therefore, magnification power and resolving power work hand in hand. So if a microscope has a high magnification power, then it has a high resolving power, and vice versa. So that is it on the differences between a light and an electron microscope. And I want to believe that if in case this question is posed in an exam, when they ask you to give the difference between a light microscope and an electron microscope, you will be able to answer that question comfortably. Very good. Now with me here, I have a question. Okay, I have a question, and I want us to participate in doing this question. So currently, if you can see the screen, I want us to name these different parts of a microscope together. Okay, so what I'm going to do is I'm going to give you a chance to name the different parts of a microscope with me from the illustration that I have here. So if I don't know if I should allow you to volunteer, or I should ask you, like I call your name and you tell us which part of a microscope you think it is. So which is the best method? Method one, where I give you a chance to volunteer, or method two, where I mention your name and ask you name the part labeled A, and then you tell me the part's name. Feel free, feel comfortable. If you are wrong, we will correct you, because that's why we are here. We're here to learn, we're here to improve each other so that we can become better students. So kindly type for me, which method do you want? Method one or method two? Volunteer. Okay. So Josephine is suggesting that we have a volunteer. So who is going to volunteer to name part A for us? Who is going to volunteer to name part A for us from what we've learned today? Part A. Part A ni nini? From the
I can
notes that
sit,
we had,
Mwalimu.
I'm sure if you are making some short notes, you already have those notes with you, so it's going to be easy for you. But anyone, kindly?
Mwalimu, can I try?
Yes, go ahead.
Eyepiece.
Yes, very good. That is very much correct. The answer to part A is an eyepiece lens. And we've said that the eyepiece lens has a lens that is used for magnification. Very good. Then we move on to part B. Part B. Anyone who wants to volunteer to tell us what is the name of part B? Anyone? Anyone? Name of part B?
Course adjustment knob.
Mhm. Yes, very good. It is coarse adjustment knob. Thank you. That's correct. And we say that the coarse adjustment knob is used to move the body tube across long distances. And this one is very important because it makes the specimen to appear even bigger because it moves the body tube across longer distances. Very good. So if you have called part B coarse adjustment knob, definitely part C becomes, anyone else? Part C becomes,
fine adjustment knob.
very good. It is the fine adjustment knob. Very good. Tumeelewa. Part E, anyone?
It's the
Yes, it is the arm or it is the limb. Okay. Yes, limb or arm, and we've said it holds the stage, it holds the body tube, and also it is used while moving a microscope. So part D, anyone? Part D, kindly, anyone? Part D?
Objective lens.
Yeah, very good. Part D is the objective lens. How many of the objective lenses do we have, by the way? Anyone? How many of the
3.
objective lenses do we have? 3, right? Yes, so
Yeah.
good. We have 3. We have the fine adjustment knob. We have, we have the medium power. We have the low power, and we have the high power objective lenses. Then we have part L, part F, part F. So part F is the, anyone?
stage.
Yes, good. It is the stage. And we've said this is where we place a slide containing the specimen. Na hapo tumesema kuko na clips, na hizi clips zinatumika kushikilia slide. So a slide is going to be placed here. So we place it there, and then it will be held by the clips like that. Okay. Yes. So then we have part G. What is the name of part G? Anyone? Part G,
Mirror.
anyone? Yes, good. Thank you. It is the mirror. And the function of the mirror is to reflect light onto the condenser and diaphragm. Iko hapo. Okay. And then we have finally part H, which we have said that part H is the, anyone? Part H?
Base
Base.
stand.
Yes, it is the base, or it is also called a stand, and this one provides a steady and firm support. So you see, those are the functions. They are the parts and their names. And thank you very much for volunteering and answering them correctly. So that is very, very good. Good. Let us talk about some maths right now. Saizi tufanya maths kidogo, okay? Now remember I've said, when you're using a microscope, the function of a microscope is to magnify. Okay? Magnification means to make something appear larger. So sometimes we can calculate how many times magnification has been done. Like for example, we can have a specimen like a cockroach, which we placed in a slide on a microscope stage, and then we want to know how many times will that specimen be magnified. There's a formula which we use to calculate the magnification of a specimen using a microscope. Now, I don't want you to confuse this with the formula for magnification while using a hand lens. So previously, in the previous lessons that we had, we said you can calculate magnification of a specimen using a hand lens by taking the drawing length divided by the actual length. That is what we said. But now here, we have another formula for calculating magnification while we are using, when we are using a microscope. Now remember we said in a microscope, it is only two parts that have lenses. These parts are eyepiece lens and the objective lens. Meaning, the lens on the eyepiece has a power and ability to magnify an object. And then the lens on the objective lens also has a power and ability to magnify a specimen. So therefore, magnification takes place in 2 points. The first part is at the eyepiece and the second part is at the objective lens. So therefore, when we are calculating magnification using a microscope, we say magnification is given by eyepiece lens magnification. Unaangalia eyepiece imeandikwa times how many times. Kama imeandikwa times 5 ni kumaanisha eyepiece iko na ability ya ku enlarge that specimen 5 times. And then you also go ahead and check the objective lens. Whether you are using the low power, the medium power, or the high power, it does not matter. Every objective lens has a magnification. It is usually written on the objective lens. So kama eyepiece ilikuwa times 5 na objective lens ilikuwa times 10, imeandikwa times 10. If you want to know how many times the specimen has been magnified, you take 5 times of the eyepiece, you multiply by 5 times of the objective lens, which will give you 50. So therefore we say magnification using a microscope is given by or eyepiece lens magnification multiplied by objective lens magnification. Okay, I've said magnification while using a microscope is given by eyepiece lens magnification imeandikwa times ngapi and objective lens magnification. So therefore, can we be able to do a few questions on magnification? Of course we can. So we are going to begin. So have somewhere you can write. So that we write a few questions. Now, the first question is, calculate the magnification of a specimen. Calculate the magnification of a specimen. Calculate the magnification of a specimen using a microscope. Calculate the magnification of a specimen using a microscope whose eyepiece lens. Calculate magnification of a specimen using a microscope whose eyepiece lens magnification is times 10. I've said calculate the magnification of a specimen using a microscope whose eyepiece lens is times 10 and objective lens magnification is times 3. So calculate the magnification of a specimen using a microscope whose magnification of the eyepiece lens is times 10 and objective lens magnification is times 3. So how do you do that? Of course, you take the magnification, you write magnification is equals to, you say magnification is equals to eyepiece lens multiplied by objective lens magnification. So in this case, magnification is going to be times 10 times 3. So the magnification will be times 10 times 3, which is going to be 30. Okay, times 10 times 3, which is going to be 30. Did you get that? Kindly type yes if you got that, like if you have now understood how you're supposed to do it. Yes, type yes. Very good. So then we can comfortably do another one together. Okay, I'm going to display a table for you here. This one here. So that table which I've displayed for you here, is a table showing some of the magnification of a specimen, objective length, and the eyepiece length. So what I want us to do is, I want us to fill in this table together one by one. So we begin with the first one. The first one has been done for us. Total magnification is times 20 because eyepiece length was times 5 and the objective length was times 4. So we are going to do the second one. So let's do the second one very quickly. Second one. So what is the eyepiece length? The eyepiece length is times 10. What is the objective length? Times 5. So what is the total magnification? Anyone?
Times 50.
Times 50. Very good. So times 50. Remember we said every time you calculate a magnification, you must begin with a times because it's showing how many times that has been magnified. Okay? Yes, so we say times 15. Have you do the second one? We have the eyepiece length of magnification as 10. We do not have the objective length, but we have the total magnification as 100. So how do we get the magnification? You take the total length, the total magnification, you divide by 10. So that one cancels, that one cancels. So therefore, what is going to be the objective length magnification? Anyone?
Times
Now
10.
what has happened? Sorry?
Times 10.
Times 10. Good, times 10. So hapa tumepewa magnification, tukapewa eyepiece lens lakini hatujapewa objective lens. So what we do is, na tulikuwa tunasema magnification is eyepiece lens multiplied by objective lens. Okay? That is equals to magnification. So what we do is, we are being given magnification is times 100, but hatujapewa objective lens, but tumepewa eyepiece lens to be times 10. So we say objective lens is x. So we divide by times 10, we divide by times 10. So that one and that one cancel. So x inabaki hii side, it is equals to divide by 10, divide by 10, then x is equals to times 10. So therefore, magnification being 100 and the eyepiece length being 10, therefore objective length will become times 10. So hapa tulisema ni times 50, hapa ni times 10. How we do the other one? So magnification ni 600, objective length ni 40, eyepiece length hatujui. So we repeat what we have done. So we take 600, we divide by times 40. So what will be the eyepiece length magnification? How many times must we multiply by times 40 to get 600 as magnification? Anyone?
15. Times 20.
Yes, good. Times 15. Good. And then the last one is a straightforward one. Times 10 as the eyepiece length multiplied by the objective lens magnification. What will be the answer? 10 times 100. What will be the answer? Anyone? It's okay. Eyepiece length is times 10. Objective length is times 100. So what will be the total magnification?
Times 1,000.
Yes, times 1,000. Good. That is how we do some questions and calculations on magnification while using a microscope. Okay, so I want to believe that now hatuwezi kosa kushika ama kujibu swali ya magnification while using a microscope. So can we comfortably say that we have understood and moved from this and do something else. So if you have understood how we calculate magnification using a microscope, then you have also understood the different parts of a microscope and their functions. Type understood so that we can proceed to the next and the last subtopic of the day. Very good. Yeah, feedback is important. It allows the teacher to know that the learners are in touch with the lesson, and also ina motivate mwalimu, anaona at least anafanya kitu. So kindly someone else. I've only seen one understood. So if you have understood how to do those calculations, kindly type understood. It will be very important. Feedback is important. Only one person has understood, so the rest have not understood. Good.
We have understood, Mwalimu. Just proceed.
Sawa basi, that's good, that's good. So let's proceed to the next part. Now, every living organism is made up of either one single cell or very many cells. Okay? Every living organism is made up of either one single cell or very many cells. A very good example of an organism that is made up of very many cells ni wewe. So you are an organism that is made up of millions and millions of cells. So therefore, if we can talk about you in biology, we say you are multicellular. Multi means many. So you are made of very many cells. If an organism is made up of one cell, we say this organism is unicellular. This organism is unicellular. Uni means one. It is made up of one cell. Unicellular. Now, so therefore, is there a difference between a plant and an animal cell? Or all cells of animals and plants are the same? That's the question we should ask ourselves. And one thing you should note is an animal cell and a plant cell are not similar at all. They might have some parts which are similar, but even the way they appear, they are completely different. So they have some similar parts, and also they have some parts which are completely different. So with that, ladies and gentlemen, allow me to introduce to you plant cells and animal cells as seen under a light microscope. Because these cells are so small, we cannot be able to see them with our eyes. Otherwise, you can just look at a person and see their cells. But because they are so small and so complex, we cannot be able to see a single cell since it is microscopic. But if we observe any cell of an animal under a microscope, this is what you are likely to see under that microscope. So an animal cell looks like this. It is almost circular in shape, but it does not have a definite shape. What does that mean? That means an animal cell does not have a shape that has a particular shape that you must find it like that. So it is not definite. So sometimes you'll find that it has ridges, sometimes it is smaller, some parts have moved on the inside or something like that. So when you observe an animal cell under a light microscope, you're not going to see very many parts. The reason why you're not going to see very many parts is because a light microscope, like we said, is not powerful. It is so, actually, when you look at any specimen using a light microscope, you're going to see very few parts because it is not as powerful as an electron microscope. An electron microscope, on the other hand, will show you even things which you never thought you can ever see. Very many, because it can be able to observe and magnify and enlarge a specimen to very high powers. Okay, so when you look at an animal cell under a microscope, these are the only four parts you're going to see. So there is a cell membrane, that is the outermost part of a cell that holds everything together. Like for example, you see as human beings, we have a skin. Not unless you cut through a skin, you cannot be able to see the internal parts of a human being. But when you cut the skin, you can be able to see the internal parts of human. So a cell membrane is used to enclose every content of an animal cell on the inside. One disadvantage about a cell membrane is it is not very strong. Actually, it can rupture very easily. It can break very easily. It is not like something we call a plant cell's cell wall. So when it comes to animal, they only have a cell membrane which is very light and it can easily rupture. Then we have a cytoplasm. Cytoplasm is a fluid that is found inside an animal cell. And this fluid has one function. It creates a medium for chemical reactions. It creates a medium for chemical reaction. So chemical reactions inside the cell take place and function, they usually take place inside the cytoplasm. So if a plant cell needs to carry out maybe respiration, it will take place inside the cytoplasm. However, we have some organelles. Organelles are very small structures which are found inside a cell that perform different functions. But you cannot be able to see the organelles details using a light microscope. You can only see the organelles details using an electron microscope like we are going to see. So we said cell membrane encloses the contents of an animal cell. We have the cytoplasm which creates a medium for chemical reactions and changes. And then we have the nucleus. Any cell that does not have a nucleus is not a living cell. Just like the way we have a brain that controls anything that we do, the brain of an animal cell or even a plant cell is a nucleus. So a nucleus controls everything that takes place inside the cell. If the cell wants to carry out respiration, the nucleus will control. If the cell wants to divide into two, because cells divide, cells huwa zinaongezeka in numbers. So if the cell wants to divide, that cell is going to be controlled and the function of division will be controlled by the nucleus. So there's a question that is usually tested in an exam. Commonly, this question asks, explain what happens to a cell when the nucleus is removed. So explain what happens to a cell when the nucleus is removed. It is very simple. When the nucleus of a cell is removed, the cell dies because it cannot survive without the control system, which is the nucleus. So the nucleus controls all the functions and activities of a cell. Now, we have a plant cell as seen under an electron microscope. A light microscope, sorry. When you look at that plant cell, you will see that there are very many parts which are not similar to those of an animal cell. And that makes the difference between a plant and an animal. Plants are more complex. They have other parts that we do not have. Like for example, an animal cell only has a cell membrane, okay? But a plant cell has both a cell wall and a cell membrane. This explains why plant cells are very tough, okay? Animal cells are not tough because they only have a cell membrane. The cell membrane is so light, it can easily rupture or break. But a cell wall does not rupture, not unless you cook that cell and destroy the cell wall. Otherwise, under normal circumstances, a cell wall is not going to rupture, neither is it going to break. So that gives plant cells an advantage. Okay? Then we have a chloroplast. The other part of a plant cell that we do not have as animals is chloroplast. Chloroplast comes from the name chlorophyll. Okay? And I'm sure we all know that chlorophyll is the green coloring matter of a plant. Okay, so what is therefore found in chloroplasts? It is chlorophyll. What is the function of chlorophyll? The plants use chlorophyll for photosynthesis and the process through which they make their own food. So if a plant cell has no chloroplast, it will not carry out photosynthesis because it is inside the chloroplast where we have chlorophyll. But since animals can't make their own food, we do not have chloroplasts, neither do we have chlorophyll. Then, just like an animal cell, every plant cell has a nucleus. Okay, and we said the function of a nucleus is to control the activities of a cell. So if a plant cell is about to make its own food through photosynthesis, it will be instructed to do so by the nucleus. If anything needs to take place inside the plant cell, the nucleus control. Then we have the cytoplasm, and just like we said in an animal cell, a cytoplasm creates a medium for chemical reactions. Then we have these two parts here. We have the tonoplast and we have the subvacuole. So tonoplast encloses the subvacuole, so it prevents the subvacuole from mixing with the cytoplasm. And then of course the subvacuole, it has a very simple function. It stores sugar and salt so that it can control what is entering the plant cell, leaving the plant cell. I'm going to repeat. I've said cell wall controls and protects the internal parts of the cell, protects them, and it gives them a very rigid ability to not burst. Okay, so a resistant ability. So it's very rigid, so it does not burst. Okay, and then we have cell membrane. Cell membrane is going to actually control and actually enclose materials on the inside, like we said in an animal cell. Then we have chloroplast. Chloroplast, we said it contains chlorophyll, which is used to carry out photosynthesis. And then, of course, we have the nucleus controlling the functions of a plant cell, and the cytoplasm, tonoplast, and the subvacuoles. So when you look at those two cells like that, they have similarities and they have differences. The similarities are, as you can see, let me show you the similarities. A plant cell has a cytoplasm and so does an animal cell. So they both have a cytoplasm. Another similarity is that they have nucleus. Like you can see, they have nucleus. They also have a cell membrane, both of them. And then we have differences, tofauti. The differences are a plant cell has a cell wall and it has a tonoplast and it has a subvacuole. But when you look at an animal cell, it does not have those things. So in an exam, they can bring a question asking you to give differences between animal cells and plant cells. Okay? Give two differences between animal cells and plant cells. I want us to look at the differences between plant cells and animal cells. So just to see how we should answer that question, there you go. So we can say that in plants, plant cells are usually large compared to animal cells. Okay, so if you compare an animal cell and a plant cell, you'll find that an animal cell is smaller than a plant cell. Another difference is an animal cell has an irregular shape. When you look at an animal cell, it does not have a shape, but you can be able to tell that it is circular or oval. The shape is not well defined, it is irregular. But for the plant cell, the shape is regular. It is defined. You can be able to see the shape. It is almost oval rectangle. Okay, so when you look at a plant cell, you can see it has a rigid shape. And then we have a plant cell has a cell wall, while an animal cell lacks a cell wall. The other difference is that a plant cell has cytoplasm and nucleus which are located towards the periphery. Let me show you what we mean. Okay, so let me show you what we mean. When you look at the nucleus of an animal cell and the nucleus of a plant cell, you'll see the difference. Let me show you. When you look at the nucleus of an animal cell, it is at the center, ndio hiyo, iko katikati ya cell. But when you look at the nucleus, the same nucleus in a plant cell, it is at the end of the, towards the end of the, on the side, sorry, not the end, towards the side of the cell. So we say nucleus on an animal cell is at the center, while the nucleus of a plant cell is at the periphery. Okay, periphery. So periphery means on the side. Sawa sawa. So that's a difference. So differences are plant cells have a cell wall, animal cells lack a cell wall. The nucleus of a plant cell is at the periphery on the side. The nucleus of an animal cell is at the center. Okay, and then the other difference is if you look at these two cells, you'll see that a plant cell has a chloroplast while an animal cell does not have a chloroplast. Okay, another difference is on the shape. Okay, look at the shape. The shape of a plant cell is defined if you look at it, but that of an animal cell, it is not defined. Haiko defined vile. So it can be oval, it can be circular, so the shape keeps on changing. So in an exam, one question you can get from this area is distinguish between or list 3 differences or list 4 differences between a plant cell and an animal cell. So I want to believe that you can now tell the differences between an animal cell and a plant cell. Now remember and don't forget that these are plants and an animal cell as seen under a light microscope, which means this is just but the basic. It is what we can see using a light microscope. So the question is, how will it appear if it is seen under an electron microscope? Remember we said electron microscopes have a very high magnification power. It can be able to see even the inside parts of a microscope, which you cannot be able to see. Okay, so ladies and gentlemen, if you look at a light microscope and an electron microscope, you're going to have completely different images. So this one which is on the screen is for a light microscope. So when you look or observe an animal cell under an electron microscope, that is how much you see. Okay, this is how it appears, that is its shape. And from the inside, you can see structures which you're calling cell organelles. Okay? Cell organelles are structures which are found inside an animal and a plant cell that perform different functions. So, for example, if you look at this diagram which I have displayed here, you will see you have something known as the mitochondria, you have something else known as the lysosome, something else known as the rough endoplasmic reticulum, ribosome, cytoplasm. We also have something known as the nuclear membrane. We have the nuclear pole, nucleolus, and very many other parts like the Golgi apparatus. So there's a very big difference between a plant cell and an animal cell as seen under an electron microscope. So when you look at these two diagrams, those differences are quite profound. And let me show you a diagram of a plant cell as seen under an electron microscope. There you go. So it doesn't have any difference in terms of shape, but when you look at it, you will see very many structures which are internal. The good thing about plants and animal cells, the cell organelles or those structures which are found on the inside, they perform similar functions. For example, if it is a mitochondrion like this one here, if it is a mitochondrion like the one that is displayed here, that one, the function of a microscope of a mitochondrion in a plant cell is the same function of a mitochondrion in an animal cell. So you do not have to worry about what is the function of a mitochondrion in an animal, what is the function of a mitochondrion in a plant. They perform similar functions. The only thing here is they can only be seen under an electron microscope. So those are the diagrams which you can observe of an animal cell and a plant cell as seen under an electron microscope. So, can we talk about maybe 2 or 3 parts of an animal cell and a plant cell, or what we call the organelles, and mention their functions? Now, for your information, these parts these structures which are found inside the cell, they are referred to as cell organelles. Okay, because they are found inside the cells. Good. Excuse me. Let's begin with some of the cell organelles. So let me discuss them from the diagram, and then we're going to look at them and their functions later. So we have the first organelle here, or the first internal structure of a cell here, which is known as the mitochondria. Okay, it is known as the mitochondria. Now, these are two. Ikiwa moja inaitwa mitochondrion. Mitochondrion ikiwa moja, and then zikiwa mingi zinaitwa mitochondrion. Now, what is the function of a mitochondria or a mitochondrion? Mitochondria ziko na kazi moja kwa cell. They produce energy. You see, everything that you do as an animal or anything that takes place inside a plant cell, those activities require energy. Like, for example, you need to move from one place to another. You need to reproduce at some point. You need to digest food. You need to do very many activities. All those activities will only be possible if you have energy. Where does energy come from? Energy comes from the food that we eat. When we eat food, that food gets absorbed in the small intestine. It is then transported through the bloodstream into our cells. And when that food gets into the cell, it moves into a mitochondrion. Okay? And it is from these structures we're calling the mitochondrions that energy is prepared from the food. So food gets inside the mitochondrion, and then it is broken down to release energy, and that energy can be used for other functions and other purposes. So therefore, the function of a mitochondria, or ikiwa moja mitochondrion, is it provides a site for respiration or a site for energy synthesis. It provides a site for respiration or a site for energy synthesis. Process through which food substances are broken down to release energy is known as respiration. So you see, like the way you go to the kitchen to cook, we call that cooking, preparing food and cooking. So where energy is cooked inside the cell is in the mitochondria. So zikiwa mingi mitochondria, ikiwa moja mitochondrion. Very good. Let's look at lysosome. Okay, we have this part here which you're calling the lysosome. So a lysosome is another cell organelle which you cannot see with a light microscope, but you can see it under an electron microscope. Now, lysosomes is like, you see, like the way you sweep your house. So you clean your house, you remove trash from the house, you remove dust from the house, anything that you don't need any longer in the house, you take it outside, you burn it together with trash, or you donate it to people, or you give it out to people if it's a dress or if it's a piece of trouser which you're wearing, a pair of trousers you are wearing and maybe it no longer fits you. So the function of cleaning the cell, destroying any cell organelle that is no longer functioning, or even sometimes destroying an old cell, that function is carried out by the lysosomes. So lysosomes destroy worn out cell organelles or worn out cells. That is the function of lysosomes. And they like testing this in an exam. Lysosomes and mitochondria, they are very, very commonly tested in an exam. So, mitochondria and lysosomes, very common, note that. So, tumesema mitochondria provide site for energy synthesis or respiration, and lysosomes destroy worn out cell organelles, or even sometimes destroys the entire cell. So let us go ahead and talk about ribosomes. Wacha kwanza turuke smooth endoplasmic reticulum, but I want us to talk about ribosomes, okay? Ribosomes. So these are the ribosomes here. Now if you look at the ribosomes, you're going to see that they are attached to H-looking structures. So these dark parts here, hizi parts ziko dark, zinakaa ziko shaded. That is the ribosome. Okay? Hii ndio ribosome moja, hii ni ribosome ingine, that's another ribosome, that's another ribosome, that's another ribosome, that's another ribosome, another ribosome, another ribosome. So all those dark structures here are known as ribosomes. What are the functions? What is the function of a ribosome? It is very easy. A ribosome synthesizes protein in the body. A ribosome synthesizes protein in the body. Now, what do we mean by a ribosome synthesizes protein? You know, someone will ask me, but si mwalimu, we take proteins in the body. So why do we need to synthesize and prepare? Synthesis means it produces proteins. Now, let me explain. You see, when we take proteins, there are two types of proteins. We have protein type 1 and we have protein type 2. Type 1 proteins are proteins from animal proteins, and type 2 are plant proteins. So when you take beans, which is a source of protein, the proteins which are in beans are not the kind of proteins which you have in your body. So therefore, proteins get digested in the digestive system to produce amino acids. Then your body will take amino acids into the body and use those amino acids to make the exact proteins that your body needs. So haukulangi protein inaenda direct ikiwa protein kwa cells. No, it is digested into amino acids, and then those amino acids are going to be used to form proteins which your body requires. So when we say that ribosomes are used in protein synthesis, we mean they combine amino acids and form proteins which you need in your body. Are we together? And then of course we have the cytoplasm. Cytoplasm is another cell organelle, and we said that cytoplasms have one function. They provide a medium for chemical reactions. Okay, so even a reaction needs to take place inside the cell, it takes place through in, not through, but in the cytoplasm. Very good. So on this side here, we have other cell organelles like the nucleus, nucleolus. Now, nucleolus is found inside the nucleus. Remember, we have said nucleus control the functions of a cell. So without the nucleus, we are not going to have cell functioning correctly. So cells only function correctly if they have a nucleus. Now, inside the nucleus, we have nucleolus. Then we have chromatin. We are going to see what is chromatin. And then we have smooth endoplasmic reticulum and rough endoplasmic reticulum. What is the difference between rough endoplasmic reticulum and smooth endoplasmic reticulum? It is very simple. When you look at the smooth endoplasmic reticulum, these ones here, let me show you. These ones there, those ones, they do not have structures that are connected to them. Like for example, look at the rough endoplasmic reticulum. They have structures that are shaded, that are surrounded, that have surrounded the rough endoplasmic reticulum. These shaded parts here, which we have called the ribosomes, because these are the ribosomes, okay? Ribosomes are the shaded parts of the rough endoplasmic reticulum. Na tumesema ribosomes prepare or they manufacture proteins, okay? So when they form proteins, those proteins are going to be transported across the body by the rough endoplasmic reticulum. So kazi ya rough endoplasmic reticulum ni kutransport. So once the proteins have been synthesized in the ribosomes, they are packaged in the rough endoplasmic reticulum. Unaona venye ni refu refu, they transport those proteins across the body, and the proteins are used in building, they get used in growth and very many other activities. But ukiangalia smooth endoplasmic reticulum haiko shaded, it is smooth, it has nothing attached to it, hence the name smooth endoplasmic reticulum. Now what is the function of a smooth endoplasmic reticulum? Very simple. Smooth endoplasmic reticulums, they package and transport glycoproteins. Okay, so they package and transport substances in the body. Sawa sawa. Now I know this information can be too much to handle at some point, but I want us to not forget three parts for now, three organelles. The first cell organelle that I don't want you to forget is mitochondria, because it is actually highly examinable. So mitochondria, we've said it is a cell organelle that synthesizes energy. Then we have lysosomes, and lysosomes, we've said, they perform the function of cleansing the cell. So they destroy worn out cell organelles. Kama hii mitochondria iko hapa juu ikiacha kufanya kazi, lysosome inakuja ina destroy. Hii cell yote ikiwa haifanyi kazi venye inafaa, the lysosome destroys it. So lysosomes are used to destroy worn out cell organelles and worn out cells at some point. So number one, usisahau mitochondria. Number two, usisahau lysosome. Number three, ribosomes. Okay, ribosomes, we've said they synthesize proteins which are used in the body for the process of growth and development, reproduction, and many other functions. And then of course you'll not forget the nucleus, because it controls the function of the cell. Good. So nimesema usisahau mitochondria, usisahau lysosome na usisahau ribosome tafadhali. And then during the next lesson we'll talk about the other cell organelles and their functions. So not unless there's a question, that marks the end of the lesson today, and we have learned quite a lot. We've learned about the differences between plants and animal cells. We've also learned about microscope and parts of a microscope and their functions, and we've also learned how to do calculations using a microscope. So if you have understood what we have learned today, I'm very happy for you, and I want us to continue the same way. If there's a question, you can ask now, and of course I appreciate the feedback. It's my I'm happy to hear that we are progressing and we are understanding what we are doing, because it lays a very good foundation for the next step, which is going to be Form 2 work and Form 3 work. So with those very many remarks, if there is no question, I want to wish you a wonderful, wonderful night, a good night, and see you tomorrow in the morning for the other lesson. Okay? Yes. So if there's no question, have yourselves a blessed night and see you tomorrow, God willing. If there's a question, you can ask. Thank you. Well, looks like there's no question, so have yourselves a blessed night. See you tomorrow in the morning, God willing. Kwaherini. Good night to you too.
Excuse me.
Yes, yes, Kate.
I have a question.
Mm-hmm.
You said that light microscope can be used to observe the dead object and the live, and electrical microscope deals with the dead only.
Yeah.
Now my question is, when you go to the hospital, like
Uh-huh.
they take some sample, which I
Mm-hmm.
believe they use electrical microscope. So the
Mm-hmm.
sample from our body,
Mm-hmm.
are they dead or alive?
What happens is before they work on that sample, if it's a stool or something, they sterilize that particular stool. So if they ask you for anything, blood, stool, or even saliva, first of all, it gets sterilized, okay? Because also when they are working with the specimen, they cannot risk coming into contact with those disease-causing microorganisms, because it would mean they would contract the disease. So therefore, they sterilize it before they even work on it. So they'll be observing dead specimen from whichever sample they have collected from you. But they must sterilize it so that they can be able to work with it. So is that understood?
Yes. Thank you.
Thank you. Well, any other question? Okay, good. If there's no any other question, kwaherini. Have yourselves a good night.